Introduction. Welcome, medical students, to today's lecture on the fascinating topic of biochemistry. In this comprehensive script, we will explore the diverse roles of various coenzymes and cofactors in redox reactions, fatty acid metabolism, 1-carbon metabolism, electron transport, and many other essential biochemical processes. Through an in-depth analysis, you will gain a deep understanding of the structures and functions of these molecules. Let us begin. In summary, flavin adenine dinucleotide, FAD, and flavin mononucleotide, FMN, are important coenzymes involved in redox reactions. They contain a flavin ring system linked to an adenine moiety through a ribofuranose ring. FAD and FMN serve as oxidizing and reducing agents in electron transfer reactions, participating in processes such as the TCA cycle, fatty acid oxidation, and the electron transport chain. They can accept and donate electrons, enabling them to be involved in crucial redox reactions for energy production and metabolism. In fatty acid metabolism, two important coenzymes are coenzyme A, CoA, and acyl carrier protein, ACP. CoA activates and transports fatty acids, and consists of a nucleotide, pantothenic acid, and a reactive thiol group. ACP is found in fatty acid synthase systems and carries the growing fatty acid chain during synthesis. ACP possesses a phosphopentate group that serves as a flexible arm, shuttling the fatty acyl chain between enzymatic sites. Both CoA and ACP play vital roles in ensuring the proper metabolism of fatty acids. In 1-carbon metabolism, tetrahydrofolate, THF, and S-adenosylmethionine, SAM, play pivotal roles in carrying and transferring 1-carbon units for biosynthetic and regulatory processes. THF is a multifunctional coenzyme that acts as a carrier of 1-carbon units in various forms, including methyl, formyl, and methylene groups. It consists of a pteridine ring, p-aminobenzoic acid, and a glutamate moiety. THF accepts 1-carbon units from different donors and transfers them to various acceptors, aiding in the synthesis of nucleotides, amino acids, and other important biomolecules. S-adenosylmethionine, SAM, serves as a methyl group donor in a wide range of methylation reactions. SAM is derived from the amino acid methionine and functions as a versatile methyl carrier. Upon donation of its methyl group, SAM is converted to S-adenosyl homocysteine, SAH, which is further metabolized to recycle the methionine and regenerate SAM. Thus, the balance between SAM and SA is crucial for maintaining proper methylation patterns and overall cellular homeostasis. Nicotinamide adenine dinucleotide, NAD+, is a coenzyme that plays a fundamental role in redox reactions as an electron carrier. It is composed of two nucleotides, namely nicotinamide mononucleotide, NMN, and adenosine diphosphate, ADP ribose. In its oxidized form, NAD+, this versatile coenzyme accepts electrons and hydrogen ions, H+, from substrate molecules, yielding its reduced form, NAD. This redox cycle enables NAD+/NAD to act as an electron carrier in various metabolic pathways, including glycolysis, the TCA cycle, and oxidative phosphorylation. By accepting electrons, NAD+ gains the ability to donate them to other molecules, effectively transferring energy from one reaction to another. This process is crucial for generating ATP through oxidative phosphorylation and maintaining the balance of redox reactions within the cell. Heme is an essential cofactor involved in oxygen binding and metabolism, primarily through its presence in proteins such as hemoglobin and myoglobin. Heme consists of a porphyrin ring, which contains an iron atom coordinated within its structure. The iron within heme can reversibly bind oxygen, enabling the transport and storage of oxygen within red blood cells, hemoglobin, and muscle tissues, myoglobin. In addition to oxygen binding, heme-containing enzymes, known as cytochromes, participate in electron transport processes during oxidative phosphorylation. These cytochromes, containing various heme groups, serve as electron carriers within the mitochondrial electron transport chain. Through sequential redox reactions, the heme groups facilitate the transfer of electrons and ultimately drive ATP synthesis. Metal ions, such as zinc, copper, iron, and magnesium, serve as essential cofactors in a myriad of enzymatic reactions. For instance, zinc plays a vital role in the activity of carbonic anhydrase, an enzyme responsible for facilitating the reversible conversion of carbon dioxide to bicarbonate. In carbonic anhydrase, zinc ions are held at the enzyme's active site, contributing to its catalytic efficiency. The zinc ion coordinates with water molecules, creating a proper environment for CO2 hydration, and subsequently stabilizing the bicarbonate intermediate. This interaction enhances the reaction rate, enabling efficient carbon dioxide transport and maintaining acid-base balance in various biological processes. The presence of metal ions as cofactors in enzymes allows for precise regulation and modulation of enzymatic activity, making them indispensable for proper cellular function. Pyridoxal phosphate, PLP, the active form of vitamin B6, serves as a coenzyme in numerous enzymes involved in amino acid metabolism and transaminations. PLP functions by forming a Schiff-base linkage with the amino group of an amino acid, creating a highly reactive intermediate. This intermediate can undergo various transformations, including transamination reactions, decarboxylation, and racemization. Transaminations are particularly crucial in amino acid metabolism, as they facilitate the interconversion of different amino acids and the synthesis of non-essential amino acids. PLP acts as a carrier and catalyst, shuttling amino groups between amino acids and acting as a cofactor for aminotransferases. The ability of PLP to facilitate these reactions is crucial for maintaining proper amino acid homeostasis, supporting protein synthesis, and participating in neurotransmitter metabolism. Retinol and retinoic acid, derivatives of vitamin A, serve as cofactors in vision and nuclear receptor signaling. In vision, retinol participates in the visual cycle, which involves the regeneration of the visual pigment, rhodopsin. Rhodopsin consists of the protein opsin and a retinal chromophore derived from retinol. Absorption of light by the retinal chromophore initiates a series of conformational changes that result in the generation of nerve impulses, allowing vision to occur. Retinoic acid, on the other hand, acts as a critical signaling molecule in gene expression regulation. Retinoic acid receptors, RARs, and retinoid X receptors, RXRs, are nuclear receptors that bind retinoic acid and act as transcription factors. Through their binding, these receptors regulate the expression of target genes, playing crucial roles in embryonic development, cell differentiation, and the maintenance of homeostasis. This highlights the importance of retinol and retinoic acid as cofactors in vision and gene regulation, respectively. Coenzyme Q, also known as ubiquinone, is a vital component of the electron transport chain, ETC, involved in oxidative phosphorylation and ATP production. Ubiquinone is a lipid-soluble molecule with a long hydrophobic tail and a quinone head group. It is ubiquitously present in the inner mitochondrial membrane and acts as an electron carrier between complexes 1 and 2, NAD dehydrogenase and succinate dehydrogenase, respectively, and complex 3, cytochrome BC1 complex. Through its ability to accept and donate electrons, ubiquinone shuttles electrons from complex 1 or 2 to complex 3, ultimately transferring them to cytochrome C and supporting the synthesis of ATP in the process. Thus, COC acts as a crucial link between electron donors, NAD or FOD2, and the final electron acceptor, oxygen. The molybdenum cofactor, MoCo, is an essential cofactor in various enzymes involved in sulfur metabolism. MoCo contains a molybdopterin molecule, complexed with a molybdenum atom and coordinated with sulfur atoms. This complex serves as an active center for enzymes such as sulfite oxidase, xanthine oxidase, and aldehyde oxidase. Sulfite oxidase, for instance, catalyzes the conversion of toxic sulfite, produced during the breakdown of sulfur-containing amino acids, to sulfate. MoCo plays a crucial role in accepting electrons from sulfite and transferring them to an electron acceptor, ultimately oxidizing sulfite to sulfate. By participating in sulfur metabolism, MoCo ensures the proper detoxification and utilization of sulfur-based compounds, playing a vital role in maintaining cellular homeostasis. Biotin is a cofactor involved in carboxylation reactions, primarily serving as a carrier of activated CO2. In carboxylation reactions, biotin is covalently attached to a specific lysine residue within biotin-dependent carboxylases. The activated CO2 is then transferred from biotin to target molecules, such as acetyl-CoA or pyruvate, enabling the addition of a carboxyl group. An example of a carboxylation reaction involving biotin is the conversion of pyruvate to oxaloacetate by the enzyme pyruvate carboxylase. Biotin's flexible linker allows the transfer of the carboxyl group between distinct active sites within the enzyme, promoting the overall reaction. By acting as a carrier of carboxyl groups, biotin facilitates essential biosynthetic processes, including fatty acid synthesis, gluconeogenesis, and amino acid metabolism. Iron-sulfur, Fe-S, clusters are cofactors found in a diverse range of enzymes involved in electron transfer reactions. These clusters consist of iron atoms coordinated with sulfur atoms from cysteine residues and occasionally inorganic sulfides. They exist in various forms, including cubane-like clusters and iron-sulfur clusters bound by protein residues. Fe-S clusters serve as electron carriers, accepting and donating electrons as part of redox reactions. For example, they participate in electron transfer within complex I, complex II, and complex 3 of the electron transport chain. Through their ability to shuttle electrons, Fe-S clusters facilitate the formation of proton gradients and ATP synthesis during oxidative phosphorylation. Moreover, Fe-S clusters also play a role in nitrogen fixation, DNA repair, and regulation of gene expression, highlighting their versatile importance in biological processes. I hope you find this video helpful in understanding the importance of coenzymes and cofactors in biochemistry. Remember that these molecules have essential functions in various biochemical processes, such as catalyzing reactions and transporting electrons. We have discussed the structures and roles of several coenzymes and cofactors, including FAD, CoA, THF, NAD+, Heme, metal ions, PLP, retinol, COC, MoCo, Biotin, and Fe-S clusters. These molecules are crucial for maintaining the balance of metabolic pathways and overall cellular function. By comprehending their properties and functions, we can better grasp the complexity of biochemical reactions and their implications for human health and disease. Thank you for watching.